Hello, everyone. Good morning, good afternoon, good evening, depending upon the time zones where you are. Welcome to the 9th webinar of ONWARD. I know you are all familiar with what ONWARD is now, but ONWARD, Open Network for Water-Related Diseases, is funded by GCRF with Dr. Milton Campbell and Dr. Shubha Satyendranath as lead investigators. And we aim to build a network of scientists and students working on the various aspects of water-related diseases in different disciplines in order to develop our knowledge so as to forecast outbreaks of water-associated diseases and risk mapping. So today we have with us Dr. Katrina Campbell, Professor, Food Security and Diagnostics, Institute for Global Food Security, School of Biological Sciences at the Queen's University, Belfast. Her research focuses on the examination of food systems for the identification and recognition of known and emerging threats, including fraud within the food supply chain, from environment to farm to fork to waste, and to determine any consequential effects to plant, animal, and human health. Her research also involves examining the impact of the effects of contamination and the implementation of these new diagnostic tools in the food supply chain to all stakeholders, including academia, industry, policymakers, and consumers. A chairperson and member of various international societies, Dr. Campbell is also the author of more than 120 publications with an H-index of 39 and extensive network within 8 food and EU projects. So, welcome, Katrina. Today, she'll speak on the topic, water monitoring, diagnostic tools for the future. The floor is yours, Katrina.
Thank you very much, Nadini, for a lovely introduction. So yes, I'm Katrina Campbell I'm a professor in food security and diagnostics at the Institute for Global Food Security at Queen's University in Belfast. And I'd like to talk about diagnostic tools for the future and those that can be used in water monitoring. In relation to the talk, what I would like to talk about are marine toxins and toxins in the marine environment, the problems that they cause, some of the current detection methods, the upcoming array platforms, multiplex arrays, electrochemical screen printed electrodes, and then looking at the future outlook for different diagnostic platforms and what my perspectives are in terms of what we should be doing in the future to improve water monitoring and quality control. So marine toxins then are phytotoxins, and these are toxins produced by algae, and algae such as dinoflagellates or diatoms. You can see in the electron microscope images, they're very small micrometer sized organisms, and these organisms form algal populations that can form visible dense concentrations of cells or blooms. And as you can see then in the picture in Clovelly Beach in Australia in 2012, a nice red tide bloom that has formed from the accumulation of some of these dinoflagellates. In terms of the dinoflagellates, the conditions flavouring this bloom growth include water temperature, sunlight, competing microorganisms, nutrients such as phosphorus, nitrogen, and microelements, but also wind and the direction of the currents are thought to be involved. And for these particular toxins, then they can enter the food web by accumulating in shellfish and fish, and on eating the toxins can cause severe illness or death. And depending on the toxin that might be consumed would depend on the different syndrome that you would experience. For example, the likes of oxalic acid, one of the diuretic shellfish poisons, would give you symptoms of diarrhea and vomiting. For domoic acid, it would be an amnesic shellfish poison, and the likes of domoic acid then would cause symptoms of amnesia and illness, as the name suggests in terms of its grouping. And then for saxitoxin, also known as a chemical weapon, This would very much be involved then as a paralytic shellfish poison and cause symptoms such as paralysis and even death. So these particular marine toxins do have to be monitored in our environment to ensure that we are preventing illness or public health concerns, not only in our water and recreational waters for water bathing, but also then in our food supply systems. You can see here then the different groupings of the toxins, the poisoning syndrome that they cause. I've mentioned already diuretic shellfish poisoning, amnesic shellfish poisoning, and paralytic shellfish poisoning. There's also neurotoxic shellfish poisoning caused by a number of emerging concerns or emerging toxins in Europe. And there's also ciguatera fish poisoning. And ciguatera fish poisoning is thought to be one of the most problematic classifications of infectious diseases for the future that we need to be concerned about in our marine environment, whereby it's believed that possibly in the future we may not be able to obtain certain fish without actually the fish containing ciguatoxin. You can see also the toxin that's produced from these particular, or the cause of these particular poisoning syndromes. You can see the phytoplankton species that produces the toxin, the vector, and the vector in a lot of cases would be shellfish and fish. And you can see some of the short-term health consequences that are known. A lot of those short-term health consequences actually were involved then in the naming of the poisoning syndrome, which affected human health. But when we look at the long-term health consequences, we are less certain of what these particular toxins do to us on a long-term basis over a number of years or decades, perhaps, if we would be consuming these particular toxins even at low levels. Some are thought to be tumour promoters, such as oxalic acid. Some are thought to lead to recurrent symptoms, such as chronic depression and flu-like symptoms. And others then are thought to really affect memory deficit in the long term, perhaps even leading to seizures, coma, and death. So they are quite a concern, right across the globe in terms of their occurrence. And with climate change, they're actually believed to be increases in occurrence of these particular blooms, which produce these particular toxins. And in terms of climate change, it's also thought and believed that these particular organisms are able now to migrate to other areas which are now warmer in terms of the sea temperature, where they can proliferate and develop. So they are very much an area of concern, and particularly around our water environment. In terms of these particular toxins, obviously from my background in food, I was interested in food monitoring, perhaps before being interested in the water monitoring. And how this sort of arose was part of a logistical nightmare in terms of the incidence of particular paralytic shellfish toxins occurring in Tasmania, just off the coast of Australia. You can see on the map magnifying glass, and the magnifying glass here shows where the shellfish beds are in relation to the coast and the island, and also the main city in Hobart. The shellfish are grown in these long ropes, and they filter feed on the dinoflagellates and diatoms. They don't appear to affect the shellfish at this point to any great extent, but they will accumulate in the shellfish for some period of time while the bloom is occurring and for some time after. And then they will naturally depurate out of the shellfish again with coming and going of different tidal movements. So for this, it was pretty much a logistical nightmare. Samples were taken from the shellfish beds. These were transported then to Hobart in terms of the city. And then they were transported by air to Sydney and Wellington in New Zealand for testing. And testing for marine biotoxins is regulatory to ensure that the produce is good to eat. Now, at the same time as they took a sample for testing, there was a large sample or a large harvest that went to China and Japan. And the turnaround time on testing can be close to a week. And the water conditions do not remain static. So basically, when the samples had got to Japan in 2012, this management system cost the Australian industry 23 million Australian dollars in recalls from contaminated batches going to Japan. And when we talk about contaminated batches, we talk about shellfish contaminated with so much paralytic shellfish toxin that one mussel would actually kill a person. So it was quite a serious incident. And it didn't just shut down the shellfish industry in Australia and Tasmania, it shut down the whole shellfish industry in Australia. So it was a major concern. So what they wanted to do was to look at different methods of detection and different methods of detection that could be used on site, rather than having to send samples away to Sydney or to Wellington for analysis. In terms of the situation, we had a similar incident then off the coast of Shanghai in China, and you can see with the red circle some of the islands, and you can see in the picture the beds and beds of shellfish production. And similar logistical nightmare where these samples are transferred by boat back to Sushan, and then they're tested there by the CDC in terms of their toxicity. Now, there's a variety of diagnostics that perhaps could be used. We were looking for black box, easy to use technologies for the industry, but the logistics of shellfish production can be a real challenge. And it's not only a real challenge in the likes of Australia and China, the UK and America also have similar challenges in terms of testing to verify the presence or absence of these particular toxins. So when we look at seafood safety monitoring, how it used to be performed was a little bit barbaric. We had a reference method, which is the mouse bioassay, and the death or illness in the mouse following injection of shellfish extract meant then that shellfish extract was toxic. So if the mouse took ill or the mouse died, it meant the shellfish was not good to eat. In the European Union, it was deemed fairly unethical, and there was a lot of research and a lot of money put into place to remove the mouse bioassay. It was also deemed for some of the toxins to be a little bit unreliable because it had interfering matrix effects. And then we looked at the laboratory-based analytical methods. And these are the methods that would be used in the laboratories in Sydney, the laboratories in Wellington, and also in Sushan near Shanghai. And these methods tend to be highly expensive equipment. They require trained experts to use. They're not really suitable at shellfish sites. And to date, we haven't been able to get the likes of high performance liquid chromatography or mass spectrometry onto the boats for rapid analysis. So again, this is a challenge. So when we looked at the different methods of monitoring for seafood safety, we looked at the biological methods, whether they be mouse bioassays, cell-based assays, and looking for changes in morphology, or receptor-based assays where the mode of action was known, and we could monitor the toxicity from the interaction of the toxins with certain receptors. We also then had the chemical methods, which started to become much more prevalent in Europe just after 2010 to be used. And the chemical methods such as high performance liquid chromatography with UV detection, fluorescence detection, or mass spectrometry detection, these were very much high-end analytical laboratory methods. They were a challenge in terms of, for a lot of the marine biotoxins, we don't have all the reference materials or analytical standards. So there was still a challenge in using some of these methods to ensure water quality and also food safety monitoring. So we started to look at some of the biochemical methods, and these biochemical methods being ELISA, lateral flow devices, and different biosensor methods or technologies. And for these particular methods, they were based on antibodies, and they were immunological assays, and they required low technology. There was different formats of how the immunological assay could be put together. We could have a direct ELISA, whereby we would have an antigen immobilized onto a 96-well ELISA plate, for example. We would have an antibody that recognized that antigen, and we would have that antibody labeled to provide a signal in terms of its detection. We would have an indirect ELISA where we would have the same setup, but then we would have a secondary antibody that was labeled. We would have a sandwich ELISA, which would be a capture antibody. The capture antibody would capture the toxin. There would be a secondary antibody to capture the other side of the toxin, and then another antibody then that is labeled to show the signal in the assay. And then we would have inhibitor antigen alongside a competitive ELISA. And these different principles of immunoassays were applied to different biosensor technology. Because the toxins are relatively small molecular weights, the sandwich ELISA wasn't the most appropriate, because usually that method is used for large molecular weight proteins. Depending on the assay format, the presence or absence of a test line will indicate whether the target antigen is present in the sample if we're using lateral flow devices, or if we're using ELISA, whether or not we get a signal in terms of change of color, compared to if we don't see a change in color. Visual interpretation of results should be possible, and some reader systems will allow for semi-quantitative determination of the amount of toxin actually present within a sample. So I want to show you some of the methods. So initially we set out to develop them methods for marine biotoxins. We set out to develop lateral flow assays, and I think after COVID, everyone knows what a lateral flow assay is. The only difference between this lateral flow assay and the COVID lateral flow assay is that when you get 2 lines on this particular assay, it is negative. And when you get 1 line on the test line, or the control line, sorry, it is positive. Unlike COVID, where when you have the 2 lines, it's positive, and when you have 1 line, it's negative. So what we wanted was a test that we could use on the shellfish beds. And in terms of the shellfish, these were actually sentinels for water quality. We could determine some of the water quality for these marine biotoxins from the particular phytoplankton through monitoring the shellfish. And we extracted the shellfish using a very simple method, and then we placed the shellfish into a cage-like bag system. And this bag had a mesh filter in the middle, whereby we were able to homogenize the sample using a roller, a bit like a paint roller. And then we were able to pour off the extract and leave the tissue behind. We're able to monitor using different lateral flow technology, and we were able to look at different concentrations of toxins. And you can see here in this particular image, when we have 2 lines, it's a negative. And as we increase the toxin concentration, we start to see the diminishment of the test line. The control line will remain, and the control line will show that the test is working. The diminishment of the line on the test line shows an increasing concentration of toxin. Now, to remove any ambiguity, a reader was brought into play to be able to read these particular devices, and relative to a quality control method built into the reader could determine whether the sample was positive or negative relative to the threshold for that particular toxin. So we set about developing methods for the amnesic shellfish toxins, moving some of the methods we had developed in ELISA over to lateral flow devices. Then oxalic acid and binophysis toxins, and then we developed methods for the paralytic shellfish toxins. Now, this particular toxin group has about 57 different analogues, all of which are important in terms of toxicity. Some are much more potent in toxicity compared to others, perhaps even by 1,000-fold. And this is where the reader became quite important, to remove any ambiguity by eye as to whether there was a line present or absent. And the reader then, if we design the method appropriately, the reader can actually give a method of quantification. So we set about to develop the method, and then we did a comparative across all the different methods that were commercially available for these particular toxins, and we compared it with the profile of toxins that we would get then in Australia. We undertook what was called a single laboratory validation of the method, and this was a single laboratory validation following AOAC guidelines. So we wanted to make sure that when we validated the method, we had validated to accredited standards in the laboratory. We then had an interlaboratory study where we had 17 different laboratories involved right across the world, including Australia, UK, Europe, China, and America. And in this particular study, it was proven that we could use the test for oysters as a regulatory method, and for mussels, it could be used by the industry as a quality control method. So it was something that actually became very important then to the industry going forward, particularly the industry in Australia and then the industry in China to validate their produce to make sure it was fit for human consumption before harvest. It became very much a sustainability tool, because if harvest was performed and the shellfish were deemed to be toxic, they had to be destroyed. However, if the shellfish were tested on the beds, they could remain in the water, and when the tidal movements changed, the toxins would then depurate back out of the shellfish. So, and the produce then did not have to be destroyed. So it was very much a sustainability tool where it could be used as point of sight testing. As I mentioned, we were able to look at different concentrations of toxin, and in determining different concentrations of toxin, then we could actually use the reader to provide a quantification. So we could quantify the amounts of particular toxins then that were found in the samples as well. But of course, we very much left the industry holding 3 different lateral flow tests. Okay, that's not such a problem. They were relatively inexpensive compared to the mass spectrometry, high analytical methods. But we wanted them to try and move forward and think of a one test fits all solution. And in doing so, we had to adopt the likes of 3D printing technology. And with the 3D printing technology, then we were able to print spots. And you can see this is the printer. It goes along a membrane strip, and we can actually print spots onto a particular membrane. We were able to apply a different reader, and you can see the size of this reader relative to an apple. So it very much fits into the palm of your hand. And one of the benefits about this particular reader is that it could read multiplex lines. So we set out to develop a method that would detect amnesic, diuretic, and paralytic shellfish poisoning toxins simultaneously. So you can see 3 different lines on the device, and also the control line to show that the test is working. It works as an indirect competitive inhibition assay. You can see the structure of the toxins are all very different. But you have to remember these are families of toxins, and we wanted to make sure that the antibodies we developed could pick up the actual family relative to their toxicity. So we actually developed this particular test and proved that it worked, and we published it just this year in Environmental Science and Technology. Now, that's for 3 different toxins and toxin groups. I showed in the table there were a number of other toxins or emerging toxins, and some concerns we have is around the spatial distribution of lines, and whereby a membrane strip used in a lateral flow device has only so much spatial orientation, or that so many lines can be put within the device before it would become crowded. So we've started looking at doing spotting and developing spots as part of our array. And of course, with this spotting array, we then have another reader, and this reader is bespoke from the printer company, Cinean, and it also then very much connects to a mobile phone device. So you can see there, you've got your computer technology in your hand with your mobile phone, you've got your reader to read the device, and this is a work in progress for us in terms of adding further toxins into a single multiplex assay. So kind of where we went from paper diagnostics or lateral flow technology was looking at different biosensors. And when you think about a biosensor, it's when an analyte reacts with a bioreceptor. And that bioreceptor might be an antibody, it might be an aptamer, it might be quantum dots, or it might be different proteins or DNA. And that interaction between the bioreceptor and the analyte is measured by a transducer. Some form of signal is given off that is measurable. And that transducer may be optical, electrochemical, mass acoustic, or thermal, and you will get a measurable signal for data processing. Now, Andy Turner defined a biosensor as an analytical device incorporating a biological or biologically derived sensing element, either intimately associated with or integrated within a physiochemical transducer. The usual aim is to produce a digital electronic signal which is proportional to the concentration of a specific analyte or group of analytes. So we looked at the key parameters that we would have to think about in terms of these particular toxins. We were going to develop biosensor methods. We were thinking about the sensitivity and the working linear range, what the limit of detection needed to be, limit of quantification, how precise and accurate they needed to be, the response time, their stability, the working lifetime, reproducibility, and selectivity. These were all the validation parameters that we had already performed for the lateral flow devices, both in-house as a single laboratory validation, but also as an AOAC interlaboratory study. So these validation parameters were still very important, irrespective of the biosensor technology. We looked at a particular diagnostic platform called Biodiagnostics at the time, and the aim was to produce a lower cost platform offering low cost analysis, simplicity in use, highly specific single target analysis. We were especially looking at multiplexing and multiple target analysis. We were looking for bespoke sensitivity for certain toxins. It had to be robust and it had to be field deployable. We still wanted to be able to use this particular device for water quality monitoring. And of course, the scientific know-how was built into a cartridge, just in the same way as the scientific know-how was built into the lateral flow device. And again, this particular cartridge, we would use different immunoassay formats and would be used to develop a test. So for an end user, they didn't need to know what was going on inside the cartridge. They just needed to know how to read the result. And what we called this technique was almost like a second generation lateral flow device. It could print a microarray of up to 100 spots. And of course, each different spot could be a different toxic assay. So we very much looked at it as a smart diagnostic tool, being specific, multiplex, accurate, real-time analysis that could be done in the field or at point of site or point of care. We used the again to print antibodies and coding antigen reagents onto the cartridges. These antibodies had been produced at Queen's University in Belfast, and we had done a lot of work in validating those reagents, and the 3D printing onto the cartridges and the building of the cartridge then we had performed here at Queen's University in line with the company for this reader. So for this particular methodology, it was important to keep, the sample preparation and all the preparatory steps for the sample simple. And it was to allow the likes of food business operators or environmental health officers to be able to perform the testing. It was to offer a simple device requiring minimal sample preparation through either simple fluid application, water, blood, milk, or juice, or dissolution of solid foods and buffering reagents, such as shellfish, for example. We add the sample to the assay buffer, we transfer it to the cartridge, and we read after 5 or 10 minutes, depending on the assay. And of course, it's like a microarray. So this time we were able to multiplex saxitoxin, oxalic acid, domoic acid, and we added into emerging toxins, cylindrospermopsin and microcystin, somewhat more familiar with in freshwater systems, but they are emerging in marine environments. So we thought we would look at this particular system. And depending on the complexity required, it could be qualitative with a yes/no answer or quantitative to provide a concentration. So it was an indirect competitive inhibition assay where we had toxins immobilized onto the cartridge surface. The sample and antibody are mixed and added to the cartridge. There's the incubation with the secondary labeled antibody. When we put the cartridge into the reader, the light's directed to the bottom of the cartridge. It allows excitation of the fluorophores of the labeled antibody, and the sensitive detection of binding events on the cartridge surface is observed. When toxin is present, there is reduced antibody binding to the surface. Therefore, in the array system, I'm using saxitoxin here as the example. When we had 0 saxitoxin present, we had a nice bright 4 nice bright spots. And when we had a concentration of 10 nanogram per mil of saxitoxin, you could see that those spots were clearly diminished in terms of the signal. So we looked at this for our 5 toxins: domoic acid, oxalic acid, saxitoxin, cylindrospermopsin, and microcystins. And we were able to develop nice calibration curves to be able to do water quality monitoring, looking at algae and toxic concentrations in different algae species, but also then to monitor for shellfish as well. So for the work we did on looking in water and cultures, we published that in Environmental Science and Technology again. Here you can see a comparison of the methods. So we had different algal cultures producing toxins, of the different cultures of different strains. They had a cell count and the toxin that these particular cultures produce. When we did a result with the biotechnology and then a result by HPLC or mass spectrometry, the results lined up fairly accurately. And in some cases, we actually detected more toxin with the MBio system, and we believe that this is due to the antibody being able to recognize different analogues of the same toxic family, where the mass spectrometry can't detect these because there's no analytical standards present. So that was the work we had done in cultures. So in terms of progressing away from the mouse bioassay, we had fairly much achieved this because at this point in time, mass spectrometry still isn't suitable for small boat environments. Perhaps in the next decade it might be. But we wanted to progress towards the development of an electronic mouse for toxin action limits. And for paper diagnostics for ASP, DSP, and PSP, we wanted to combine this into one nanoarray device for novel portable sensors. And of course, for smart diagnostics, there would be inexpensive procedures using multiplex diagnostics suitable for the industry and to be used on site. So it would be much more of a sustainability tool as well as a safety tool. Other elements then I'd like to talk about in terms of detection systems and technologies. Another device we looked at was around screen printed electrodes and using electrochemical techniques. Again, these are portable tools and becoming much more affordable in the future. And with these particular screen printed electrodes, we could modify the electrodes and we could look at the effects of different particles on the electrodes to improve sensitivity. And one of the benefits perhaps in the future of being able to use electrochemical devices is that you'll be able to put them onto inline systems. Instead of having to do the point of sight testing as an operator, the point of sight testing can perhaps be done as an online system that doesn't need maintained or the analysis performed. But of course, in looking at these techniques, we wanted to investigate the potential of different nanomaterials for developing the device. And we look at the potential of carbon black to improve screen print electrode performance. We also developed and validated methods using magnetic beads-based assays for domoic acid detection in shellfish, using these carbon black modified electrodes. And the benefits of using these magnetic-based particles is that we could use those to purify the toxin, and purify the toxin out of quite a dirty crude sample before it's added to the electrode and before the analysis is done. And this is work that was fairly successful and work that's still ongoing within my laboratory. Some work we did in collaboration with Barry in Monterey Bay. They were very interested in looking at remote sensing of living organisms and the toxins that they produce using an environmental sample processor. Here you can see the engineers at MBarry with the environmental sample processor in the top picture. In the bottom picture, you can see these silver pucks, and they're a bit like ice hockey pucks, but inside these pucks is the methodology for being able to do ELISA-based assays. But not only ELISA-based assays, also in situ hybridization assays. And in situ hybridization assays to be able to determine the species, but then the immunoassays to be able to measure the toxin present. And they have deployed these environmental sample processors in Monterey Bay. They've been able to monitor how they're performing using radio frequency modems and cell connectivity to the shore. And of course, with the interchangeable puck system, they've been able to monitor different assays at different time points. And here you can see one of the domoic acid toxin arrays using competitive ELISA protocol for monitoring toxin in the water environment in real time. So those are just a few of the examples of what we have done or how we have collaborated with others to develop water monitoring systems. Some of the future outlooks we have to think about are very much around paper diagnostics and the use of these paper diagnostics and also solid phase extraction diagnostics and systems, and how we can multiplex those to detect one or more items may be of relevant to a particular sample, particularly when we start to think about infectious diseases or looking at viruses in the environment. It would be much more appropriate if we could actually do multiplex analysis. The other area that's very much advancing at this particular time is around nanotechnology and nanomaterials. And using nanomaterials then of different sizes very much can be used then as different signal generators. Because if we look at gold and silver of different sizes, they emit a different colouring. And we can use that colouring to our advantage. If our lateral flow device, for example, is red, we're very much using gold nanoparticles. If it is blue, we're sometimes using latex nanoparticles. So a different colour could represent a different toxin that would be much more visible by eye. We also have to think about microfluidics, and more so than ever, we have different 3D printing technologies now very much at our disposal. And we can use these technologies to build different microfluidic systems that can be used on inline systems for monitoring toxins in remote environments, or looking at viruses or other pathogens of concern in remote environments, whether it's in freshwater or whether it's in marine environments, or it might even just be in wastewater management plants. We can also think then of engineering and remote sensing and how all this comes together. So you can see in the future to actually develop diagnostics, we need a number of disciplines to come together. We need to be multi and interdisciplinary to be able to develop systems such as the environmental sample processor that can do remote monitoring in real time. So when we think about rapid diagnostics for toxin analysis, we want them to be reliable. We want them to be relatively low cost. We want to make sure they have a low false positive rate and no false negatives. So we don't want any samples showing up negative that should be positive. And of course, more so, we have to think that the methods are safe to use. And we're more so thinking about the solvents and the extraction procedures that we use in such tests. Because if we want to put these tests into the hands of novice users or inexperienced operators, we have to make sure that they're going to be safe when they're applying them or using them. We looked at antibody-based methods, but more and more so, we're starting to look at other binders and in particular the use of aptamers. And again, the use of aptamers is very suitable for the likes of electrochemical techniques that can be used for rapid analysis. We're always looking at new biosensor technologies and applications are always needed. In a lot of cases, the biosensor technologies are put together by physicists and really they prove that the system works in either buffer, urine or sera samples. And very often they don't experiment beyond that before they would publish that they have a prototype instrument ready for use. When we think about water quality monitoring or when we think about food monitoring, of course, it's a different matrix that might need to be examined. And some of these new biosensor technologies aren't able to deal with the matrix or the high throughput required for such analysis. So these are things we need to think about for the future. We need to think about the speed of these new biosensors. We need to think about cost, the portability, that they can detect one or more things in terms of multiplexing microarrays. Remote sensing would be ideal, where we can get more data in real time that can be monitored 24/7. We also in the food world have to meet the requirements of regulators or industry in terms of setting action limits or maximum residue limits. And of course, for end product testing for release systems such as shellfish, these particular rapid diagnostics can be very, very, very useful. And of course, then we have to think about the other technology that's coming along, our computing technology, our data science technology, our bioinformatics, and also the use of mobile phones. These mobile phones can act as sensing units in their own right using the high resolution camera. They're very much a computer because they've got an operation via a touchscreen. They have a graphics processing unit. They have analysis tools as smart applications. Nowadays, they have fairly large memories and data transmission via mobile networks back to laboratories, for example, can be very useful then in remote sensing. So again, the use of the computing technology and even the use of computing technology, thinking about the Internet of Things and artificial intelligence is perhaps part of the way forward as well. So that's kind of where I want to wrap up. I've given some examples in terms of the diagnostic tools that we have used. And it's really now to take any of your questions that you may have.
Thank you, Katrina. And we do have 2 questions.
Okay.
Yeah, and the first one is by Modibo. And the question is, he has thanked you for the brilliant presentation. And he wants to know, how can groundwater heavy metal nephrotoxicity be analyzed among population in a given community?
So I think it's important that, you know, the samples are taken, you know, from, and it depends what you're looking for in that brown water in terms of the contamination and how it can be analysed. with the different contaminants that are discoloring the water. It's not necessarily that it's going to be a health concern if it's just soil discoloration, but it is something that would need to be discussed and looked at further.
Yeah, thank you for that. And we have another question. The person has not given us name. What is the advanced diagnostic tools to test furan and dioxins in water and more user-friendly in terms of low cost and high speed for the public to run the test?
Yeah, so at the moment, furans and dioxins are very much used with mass spectrometry, getting down to the pico level and femtomolar level for being able to be determined. There are new developments in antibodies whereby they are now achieving that level of sensitivity. And of course, we have to remember that these are also families of toxins. They're not just one compound or two compounds. And it's ensuring then that if we develop a future diagnostic, we look to multiplex for as many of those compounds as possible. But at the moment, mass spectrometry would be the method of choice for a lot of analytical laboratories. But there are opportunities in developing rapid diagnostics in this space.
Thank you. And the next one is by Aishwarya Robbie. She wants to know whether the levels of toxin can be measured from other marine organisms.
So yes, they could be. So using these different diagnostics, we can have antibodies to some of the other toxins. We can use receptor-based assays for some of the other toxins. And we can also use HPLC and mass spectrometry to measure some of the other toxins. My research group is currently looking at being able to develop further antibodies to some of these marine biotoxins with the right sensitivity and specificity to be able to add them to the multiplex assays that we have already developed. But yes, there's lots of opportunity in this particular space for developing new methods of analysis for point-of-site testing.
Thank you. And I think we have come to, we have one more question coming up. That is from Atia. What can be its implication in coral reef study? The question is not very clear. Hope the person meant the implication of water quality testing or toxin testing.
So I think, for monitoring our coral reefs, I think it would be important to monitor what the environment is in relation to those, to try and preserve them as much as possible. There's a number of things affecting the coral reefs at this point in time, but it would be important to continue monitoring and to improve the water quality around those because they're actually very precious.
Yeah, and maybe the harmful algal bloom tests that may be of more importance.
Yeah.
So I think we have come to an end of the questions. And if we don't have further questions, I would like to wind up because today we have a match between Brazil and Croatia. So we should not keep the audience waiting. And okay. So thank you, Katrina, for the very impressive lecture on the biosensors and the instruments about how sensitive, and what was more impressive was the simple assay regimes and the minimum sample preparation that they need. And thank you. It was a totally different topic because we have been dealing with multidisciplinary topics, and I hope that all the participants enjoyed.
Okay.
And
very much.
And a big thank you to Jose and Katerina for arranging everything as usual perfectly well in a short span of time. And again. Milton, would you like to add anything?
Just a few words, Nandini. I'd like to say good morning, good afternoon, or good night to everyone from Brazil to India. And I'd like to thank all participants registered or attending this ONWARD webinar under the auspices of the Open Network on Water-Related Diseases project, which is funded by the Global Challenges Research Fund from UK. I'd also like to thank my colleagues who agreed to collaborate with the organization of this event, especially to Katrina for accepting to give this interesting talk today. And there are our respective institutions, including the Institute for Global Food Security, Belfast, the Air Center, Mercy India, Plymouth Marine Laboratory in the UK, and of course, INPI from Brazil. And I should mention that this was the last webinar of this year, but we hope to continue counting on your participation on 2023. And Nandini, I think she is about to mention that you have already scheduled a webinar for next January, and the details of which will be released as soon as possible. And we hope that the new year is full of health, happiness, achievements for everyone. I take already this opportunity and wish you all happy holidays and a happy new year. And thank you very much. So Nandini, please.
Yeah, and as Milton suggested, yeah, we are happy to end 2022 with Katrina's lecture. We have had webinars as well as training programs on behalf of ONWARD, and ONWARD membership has risen to 350 members, which is, I suppose, a good number when a scientific forum is considered, and 350 members from around the world. And so see you again in 2023. In the second half of January, we'll be back with a new webinar. This time the speaker will be Dr. Michael Dillon, lecturer in infection and immunity at the Peninsula School of Medicine, University of Plymouth. And we'll be back. Wish you all a Merry Christmas and a Happy New Year. Thanks to Air Center, Jose and Katrina. Okay, bye. Bye, everyone.